this slide says, and I don't know if you guys had a chance to read it while you were filtering in, this presentation is going to be a little bit different from your traditional bio presentations. It's going to be about computational neuroscience. So it's going to be covering topics more in computer engineering, computer science, and electrical engineering than bio, protein pathways, that sort of thing. So please, please, please stop me and ask for questions. I know a lot of people say things like that. I actually mean it. There will be built-in times to ask for questions as well, but just stop me whenever, because I can just address the concepts as we go. So I'm going to be presenting on how to build a Drosophila eye. Drosophila meaning fruit fly. So again, quick background on who I am. Like Mr. D said, I graduated here 2 years ago. I've been at Columbia University for the last 2 years. I'm an engineering student there, majoring in computer engineering. For the last year or so, year and a half, I've been in a fruit fly lab that has been doing brain simulations. So that's what we're going to be talking about today, is how to actually simulate brains on a computer. So also afterwards, if you guys have any questions and you want to stop me in the hallway or stay after this presentation to ask about college admissions, Columbia, IRT, doing research at over the summer, anything like that, feel free to stop me, or you can just shoot me an email, which I think is still kapoor@computer.org. All right. So why does this matter? Why do we care about building any kind of virtual model of a brain in a computer? So there's a couple of reasons. This is actually a pretty big goal in bioengineering, and a very, very big goal in computational neuroscience. And the reason is because it impacts a lot of different fields. So, I mean, these are the fields. There's more than just these, but these are the main ones. What do I mean by these? Well, computer architecture is how your computer works. If we can get brain simulations working faster and better and stronger, then we can make computation better. We can make processing better. We can make the things that make our computer, the little chips inside them, run faster and be stronger. Artificial intelligence, I think, is a no-brainer. If we have brains.
Come on, artificial intelligence, no-brainer.
Fine.
That's awesome.
Also, if you guys want to stop and ask me how to best annoy Mr. D, just shoot me an email. Okay, artificial intelligence. Obviously, if we have brains and computers that can think like humans do, they'll be a lot more responsive, a lot more reactive than the stuff that we have in silicon right now. Medicine is again pretty obvious. If we have the ability to kind of take apart a brain in a computer, if we have the ability to kind of test it in a computer, we can see how things work in a way that we haven't really or don't really have access to right now. That includes a lot of neurological problems like Alzheimer's, things like that. And then my favorite, and the reason that I got into this field in the first place, was because of neurotech. Neurotech is basically the combination of any kind of technology, computers like this one over here in the front, attaching that directly to your brain. And so think Google plus neurons. And that doesn't just mean the ability to tweet with your thoughts. That also means things like neurological prosthetics. So if any of your older relatives have cochlear implants, that's an example of a neurotech prosthetic. It's something that it hooks directly into your brain, allows you to hear much better. If you do want to search the TEDx talk I gave, that was about immortality and how neurotech will eventually lead to immortality. The thought process there is if you can download a brain to a hard drive and then play it back, you've effectively created somebody living on a computer who can last much longer than our biological counterparts can. And of course, that kind of leads to the Matrix and being able to live in a virtual reality, et cetera, et cetera. So I think these are really awesome. I think neurotech is probably going to be the next big revolution in technology that we're going to see, certainly within our generation, maybe not Mr. D's generation, but definitely in ours. All right, so that's why this matters. So why have we not done this already? What are the big problems that kind of face this kind of neurological simulation? So there's two really big ones and one smaller problem, and I'll address why the third one is a smaller problem in a second. The first one is pretty obvious, right? Brains are really, really confusing. And we've done a lot of research to kind of understand how they work, and for the most part, we still don't understand a lot of it. We don't understand where thought comes from, we don't understand where memory comes from, and while we do have a better understanding of the individual building blocks that make up the brain, the individual components that make up the brain, a lot of it is still kind of shrouded in mystery. We don't really understand how input becomes output. That's the first problem. Second problem is that existing models just aren't very good. And there's two reasons why they aren't very good. The first one is that they're too specific. So you have a lab that's working on a model of a vision system. You can only do vision system processing with that. You can't expand it out. And so that becomes a problem because there's no shared system or platform that allows multiple labs or multiple groups to collaborate on and kind of build independently without bumping into each other. Yeah?
Is that a communication issue, or is that because the technology in the lab is so specific to the lab that it can't be?
It's more how the systems are designed. So the processing, and I'll kind of get into this a little bit later, like why what I'm working on is designed a little bit differently. But for the most part, what they do is that they design their code in such a way so that it can only really work for their vision system. There aren't like additional pathways that are thought about. There aren't additional components that are thought about. It's not really built with flexibility in mind.
But if it's modeled after an actual brain, shouldn't there inherently be flexibility there?
Yeah, you would think. So like, and this is kind of getting into a little bit about like how computational engineering is a little bit different than straight bio. A lot of other labs don't really think about like, okay, are we going to build in the capability for people to create the fundamental building blocks, like neurons, right? Do we build in the ability to allow other people to just kind of connect neurons of their own? Most other labs don't do this. They kind of build in the networking pathways, and they don't really allow other people to access how to network things together. Hope that clarifies some stuff. The other problem is that they don't really scale that well. So if you try and run, Windows 96 on this computer, right, it's going to still look and feel like Windows 96 because that's just a limitation of the software. Other models that exist right now, if the hardware gets better, if you get better Intel chips, if you have better graphical processing units, things like that, the software still has an upper limit. It kind of lags behind. And so as a result, what ends up happening is every time you have a new piece of hardware that comes out, you have to basically recreate the code behind the model. And that's a real pain. That shouldn't happen. So those are two really big goals, is how we can build a system that kind of addresses both of these. And then the third problem, and the reason I call it a smaller problem, is because mammalian brains are really, really big. They're really complex compared to other animal systems. 10 to the many neurons, right? We're talking about billions and billions of neurons with trillions of synapses. It's just a lot of computational power. So the reason this is a smaller problem is because if you can solve these first two issues, if you can create a system that solves those first two issues, you can kind of solve the third one implicitly, and then it's just a matter of adding more hardware and making it more powerful. But it's still worth considering that you don't start with a human brain. You don't start with the most complex thing you can think of. You start with a simpler model. So what are the solutions? What are the parameters for the software that we're looking for? How can we actually model virtual brains? So I think this first one is kind of tautological, right? Old software doesn't scale very well over hardware, so we make software that scales well over hardware. But the second two are a little bit more interesting. We want a piece of software that allows us to target specific networking functionality in the brain. What do I mean by this? Well, a lot of models that exist right now don't allow you to actually delve into what's happening in the processing. They only allow you to see what goes in and what goes out. We need a tool that allows us to actually take apart any part of the brain that we want at any point and see exactly what processing is going on in there. And the third part is it has to be easy to collaborate with multiple labs. And this is actually more of a practical issue than anything else, but it's actually still very, very important. Because the brain is just too big for one lab to tackle on its own. So you have to have a piece of software that, if you have a vision lab somewhere, an auditory lab somewhere, an oral system lab somewhere else, they all have to be able to work on the same system, on the same platform, without necessarily bumping into each other. If the vision lab makes changes to their part of the brain, it shouldn't necessarily affect the work that other people are doing, and the system should be flexible enough to allow multiple people to work on it. So that addresses these two big problems. The third issue is, okay, what animal model do we start with, right? We need an animal model that ideally has similar sensory systems to humans. Obviously one that's much less complex, but something that's similar, something that works in more or less the same way. We need an animal model whose genes and brains can be easily modified. Something that we have the tools to study very, very closely, to manipulate and do controls with, and see if our testing actually is returning the results that we expect. And all of this just kind of summarizes down to we need a model that's really well studied. So those of you guys who do IRT with the zebrafish, or who have worked with zebrafish or presented on zebrafish at all, it's a lot of the same considerations, right? Why do we use zebrafish? Well, that's why. So, introducing what I've been working on for the last year, it's a project called Nero. It's an open source project coming out of the BioNet lab at Columbia. So what is Nero? Well, Nero is an open source software for basically exactly what I've been talking about, a platform upon which to build a brain. Yes?
Sorry to interrupt, and this is probably a meaningless question to everyone but me, but are you associated, or is this working with Genelia Farms at all?
I don't think so.
Okay.
No problem. Yeah, so Nero. Open source software as a platform for building brain models. There's actually two parts to this work. The first one I think most people would recognize is Nero, right? Short for neurological, neuron, basically something to do with the brain. But the more technical people in here might recognize the second word, kernel. A kernel in the tech world is another way to say operating system. Like the Linux kernel, Windows OS X, these things are all different types of kernels. What we're trying to do here is we're trying to build an operating system upon which you can build a brain. Now what does an operating system do? An operating system basically interacts, it's the middle part between the program that's running on your computer and the hardware that's in your computer. It's the part that communicates between whatever your PowerPoint and my hard drive and my RAM and my CPU and that sort of thing. So what we're trying to do is we're trying to build an operating system, a piece of software that acts as a layer between any hardware and the code on top of it. So any simulation on top of it. So a good place to start when you're talking about brain simulations is how do you simulate a neuron, right? Because the neuron is the building block of a brain. Any model that is flexible enough, any system that is flexible enough, allows people to basically simulate their own neurons. How do we do this? So this is a neuron. Quick show of hands, by the way, how many of you guys have taken Bio 2? Sophomore Bio. Oh, cool. Okay, Mr. Cool. So what you should see here what you see here should look somewhat familiar. That's an action potential. Action potentials are basically how neurons interact with other neurons in the brain. So, a neuron is a cell. It has a cell wall, it has proteins, it has DNA, RNA, nucleus, whatever. But from an electrical engineering perspective, you can strip all of that away, and you can take this neuron and make it just this relationship. So let's analyze this graph a little bit. There's two axes, voltage and time. And you can see here this number is kind of increasing and then it loops back. That number is representative of current. So what you really see here is a relationship between voltage and current. In other words, you have the building block for basically any circuit that exists in the world. And I'm not talking about like neuro circuits, I'm talking about like silicon circuits, things that you can actually hold. So if you take this and kind of make it just this model, all that you have and what you're left with from a signal processing perspective is a relationship between voltage and current. Now, this neuron, from an electrical engineering perspective, is the exact same thing as this circuit. It's the same. This is the Hodgkin-Huxley model of a neuron. It's extremely important. These two guys, Hodgkin and Huxley, are two very prominent neuroscientists. They actually won the Nobel Prize in, I think, medicine for this model. And what they basically said was that this circuit, and by extension these four equations, have the same output, have the same voltage amplitude relationship, voltage ampere, voltage current relationship as this neuron. So they actually stripped away all the biological components and made this neuron into a circuit. Now we don't really have to worry about where these equations come from. I suggest you take a second level electrical engineering course in college if you're really interested in that. But just understand that this neuron can basically become these four equations. And if we can simplify a neuron down to equations, then we can run those equations in a computer. And that's more or less what we do. Yeah?
Are you taking into account the invertebrate weirdness of non-spiking neurons?
Yes. So there are actually, the neurokernel as a platform allows you to build different kinds of neurons. We are working with like 5 different models right now. We actually most recently built a Hodgkin-Huxley model, but there's also like Morris Lacar and other models like that. I don't know if those names mean anything to the rest of you guys, but yeah.
Cool.
So we do allow for different kinds of neurons. That's kind of a built-in functionality. Okay. So, and that's basically what we do. We take this circuit and put it onto this microchip. So what is this microchip? This chip is a 1 part of 100,000 that exists on this GPU. How many of you guys know what a GPU is off the top of your head? Okay, a few of you. So GPU is a graphical processing unit. And the reason we put these on GPUs, I'll explain a little bit more later, but basically a GPU is a chip on your computer that has a lot of really, really tiny computers, like hundreds of thousands of really tiny computers inside the chip. And each one of those tiny computers, we call them cores, can run very, very simple algorithms. For the most part, GPUs are used to render things on a screen. So, there's probably a GPU in that projector, there's a GPU in the computer. Those are the things that allow us to see output. They control pixels, they control coloration. We decide, okay, we're not going to use a GPU to deal with images. Instead, we're going to take our Nero circuit, we're going to take our replication of a neuron, and put that on this chip. And we're going to do that 100,000 times. So now we have 100,000 neurons all running at the exact same time that live on this chip. Right? We're starting to get something that actually looks a little bit like a brain. Now, these neurons aren't talking to each other yet. They're just kind of existing. So we actually need something that can connect the neurons together and actually allow them to communicate. That's where this part of the little computer comes in, called a CPU. How many of you guys know what a CPU is? Yeah, okay, most of you. Right, central processing unit. This is the main part that makes most of your computers tick. Yeah, so GPUs are where we have all our neurons. CPUs are where we do our networking. So I do want to take a quick tangent, explain why this is all important, why this is even relevant to mentioning. So for a really long time, people thought that you could only program on CPUs. All of your programming had to be done on the central processing unit. Nobody really imagined that you could use a GPU to do anything besides rendering images on a screen. That's a problem because CPUs, though they're really, really cool, are inherently limited. There's only 4 to 8 cores. Remember, each core is like a mini computer. And while each core is very, very fast, they can do a lot of really complex things, there's only 4 or 8 of them. Which means when you code something, you have to make sure that your code follows a sequence. A, B, C. You can't have A, B, and C running at the same time. That's a real problem for brain simulation because neurons are all running at the same time. It's not like they just follow in turns. So when people realize that, hey, you can hack into a GPU and have thousands of really small calculations running at the exact same time, then people also realize, and this is what the Bionet Lab is doing, that you can use this to make really, really effective brain simulations by simulating all of these neurons running at the same time. So yeah, just quickly covering what's on the slide. CPUs tend to have less cores. They tend to make up the majority of your computer. They run operating systems. They run programs. Compared to GPUs, which have thousands of cores but can only handle really small algorithms, really small equations at the same time. So this is what neurokernel looks like. Obviously neurokernel is a computer, right? So it doesn't quite look like this. But this is what the hardware component looks like. You have the GPUs running at the bottom. That's where all of your neurons are stored. And you have the CPU at the top that connects all the neurons together, allows them to communicate. All right, questions? Any questions so far? Yes?
How is the CPU helping them communicate?
So that's actually, we're going to get into higher level neuro stuff. We're going to talk about how these things are actually connected. But basically, what it does is GPUs alone can't communicate because they exist separately. They're separate microchips. And so neurons that exist within the GPUs need to be able to communicate with each other. So GPUs alone are just kind of running. It's like a car with an engine and no steering wheel, right? You need something to be able to tell it how to go. So that's what the CPU does. All right. So let's talk about how we actually make these neurons now, right? We talked a little bit about what the underlying structure is. Let's talk a little bit about the structure that's on top, the layer that most researchers will actually interact with. So this is a model of a fly brain. And you can see that there's very distinct colored portions of the brain. If you ever look at a model of a human brain, you will also see colored, most often you'll also see colored versions of a human brain. These define different portions of the brain that control different aspects of the fly body. So in the human brain, we have the amygdala, the hypothalamus, the prefrontal cortex. All of these regions are clusters of neurons that together combine to do a specific thing. And that's also true in a fly brain. So when we design neurokernel, when we want to think about, okay, how are we actually building this system? We want to think about how can we mimic this kind of functionality in the brain? How can we mimic this networking capability where distinct clusters of neurons are working together? And that is something we call an LPU, a local processing unit. So neurons in the brain all function independently, right? But we also understand that distinct connections of neurons, distinct groupings of them, can do much more powerful and serve much more powerful signal processing goals in the long term. So even though all neurons more or less look like this, have this relationship, together, when combined in the millions and billions, they do a lot of different things. An LPU is a way to basically identify and cluster off different portions of different amounts of neurons. And an LPU can be any size. It can be 100 neurons, it can be 10 neurons, it can be 1,000,000 neurons. But basically what it allows a researcher to do is cordon off any amount of neurons that they want and treat them as a separate processing unit from everything else. And for those of you who are interested in the technical aspect, this is mostly coded in Python with a little bit of C in the back end. So why are LPUs really cool? Well, these three equations are all the same. And if this is wrong, Vernesco, don't correct me on this. So these equations are all the same, but obviously these top two, they're much more nuanced than the third one, right? Everybody can see, oh, 1 + 1 = 2, right? It's really simple. There's just this whole thing in the middle just equals 1. You can simplify it down. And in the same way that you can kind of black box that component of the equation, you can treat it separately from everything else. You can get rid of the nuance and just understand the input-output relationship. In the same way, LPUs allow you to black box different components of the brain. So the brain looks a little bit more like this equation at the top, right? It's really complex. There's a lot of stuff going on. But if you don't know all the stuff that's going on, but you do know the more simplified version, you can take an LPU and replace the more complex stuff with a simplified model. Let's say, yeah?
But isn't this kind of exactly opposite what your stated purpose was?
What do you mean?
Like, so if you're black boxing part of the problem, then you're decreasing accessibility to it, decreasing the ability to edit it.
So this is more along the lines of like, different labs can work on this at the same time, and it's just like how to explain this? What I mean by black boxing, I don't mean like you're taking it out of the system. I mean that you're replacing what's originally a very complex system with something a little bit simpler for modeling purposes, and then when research catches up, you can go back in and make more complex. So for example, we don't necessarily know all of the connections that make up the prefrontal cortex, but we do know, we do have like a basic high-level overview of the signal processing stuff that happens. We know more or less when something goes in, what comes out, that sort of thing. So we can, instead of, oh this went back, my bad. So we can take an LPU and model it more simply. Instead of having all of like the millions and millions of neurons networked together in that one LPU, we can replace that with something simpler that achieves the same input-output goals.
Right, but I thought the point of this was that you wouldn't have to do that, because when you do that, you decrease the ability to see what's actually happening and access any part of it or change any part of it.
So then other people, so this is, the limit here is not necessarily access. Then once, it's more what we know. Like there isn't enough research to connect all the neurons. Once the research comes out, it doesn't matter which lab it comes out from, they can go into that LPU and change it. Yeah?
You have 5 minutes.
Cool. Okay, so yeah, so LPUs are pretty awesome. I kind of hinted at this when I was talking with Mr. D. They also allow different teams to work on brain components without affecting the rest of the brain, which is very, very important. So this is a higher level overview of neurokernel. Once we add the LPU plane back in, you can kind of see how we're constructing an operating system here. Because people who code LPUs, they don't have to worry about how those LPUs interact with the GPUs. They don't have to worry about how the software is interacting with the hardware, it just does. So neurokernel is this platform that allows people to create structures in the brain without necessarily wondering how that is going to connect directly to the hardware. And of course, the hardware considerations scale very well, allowing it to kind of be as flexible as necessary. So here's what we covered. Any other questions? Okay. So my research was about building a fly eye. I actually started the presentation like that, right? So I've been talking a lot about the background of like what I've been working on. I'm now expanding the model of neurokernel to include a working model of the fly eye. There's 2 parts that we're working on. The first part is the retina, the second part is the lamina. Retina is the same as it is in humans. It's the part of the eye that actually gets vision, right? It converts lights and photons into meaningful receptors, that signals that the brain can process. The second part is the lamina. That just sits right behind the retina. It's the second step in vision processing. You don't really have to worry about so much about what it does. Just know that it's like another step, another layer deeper into the brain that converts signals into vision processing steps. What we did, or what I did, was I hooked this camera up to a model of the retina and the lamina, this real-time input, much like our eyes work. And hooked it up to a model in neurokernel of these 2 systems. Together, those are about 13,000 neurons, 50,000 synapses between them, and this eye has about 2 billion equations. So we're running a lot of calculations at the same time. So you can kind of see why we really need to get something running in parallel, like a GPU, at the start. And you'll notice that all three of these, the input, the retina, the lamina, have outputs that go directly to the display. I actually wanted to highlight this. This is actually pretty important because it allows us to treat each component separately even while they're running in tandem. So we can see what the original input was. We can see what the retina does. We can see what the lamina does, and then we can see what the whole thing does as a group. In other words, we can break down the networking functionality of the fruit fly brain. And we tested this. This was our output. So you can kind of see this was the original picture, right? And you can actually make out what happens at each step of the vision processing. So it goes, original. This is what gets sent into the retina of the eye. This is what happens within the retina, and then what it gets output to the lamina, and then again, what happens within the lamina and what gets output there. So what we're seeing here is literally we're delving into the processing components of the fruit fly eye. We can directly see what's happening. We can break it down step by step. And I think that's really cool. I do want to highlight real quick that there was actually a biological component to this as well. It's well known that the lamina in Drosophila, they flip the polarity of light. So if something comes in as white light, it'll come out as black in the vision processing and vice versa. And you can actually see that output here. So this white component here comes out as black. This black component here comes out as grayish black. So that's something that we weren't necessarily expecting because we didn't program that in. All we did was connect the neurons in the right way and it released the same output. So discussion. Neurokernel as a platform is really ideal for building brain models because of the reasons we discussed. Modular development allows for parallel research with different labs. It scales really well with different hardware and allows step-by-step signal processing breakdown to really see what's happening in the brain. And then future plans, what we're going to be doing with this stuff. My future plans, writing a paper right now, we're going to publish it, and then we're going to be building an actual robot with this, like a little NXT bot that's going to have like basic collision detection from the input that it gets from the retina and the lamina. We're going to have it running around the lab. If I have my way, it's going to be attached to a drone. That drone will have a very high-pitched buzzing sound. It's going to fly around and annoy people. It'll be a lot of fun. So, any questions? That was basically everything I've been doing for the last year and a half. And it is 8 o'clock, so I don't know if you guys have to run off to class, but yeah, I'll be around for a couple minutes afterwards if you have any questions about anything at all that I talked about. Hope you guys had a good time.
And if you want to get a hold of him, you can email me.